Okay, well, thanks very much. I was a bit surprised when Richard invited me to come to this conference. I said, I'd really like to come. I'm going to learn a lot, but I'm not sure what I can teach you. So I'm not a statistician, and I'm not a computer scientist. I don't work in developing these methods. But I have been interested in following this literature for a very long time. In fact, when I was doing my postdoctoral research, myself and my supervisor, we started looking at this very strange, new, and bizarre statistical analysis called structural equations modeling, which for biologists was just very, very, very strange. And when I started playing around with these, I started realizing these sort of questions. Okay, well, if I get a model, are there other models that work? And just by chance, I was wandering through the library one day, and I came across a book called Discovering Causal Structure. And it was such a provocative title, I had to stop and take a look at it. And so since then, I've been interested in trying to follow up in this literature, but obviously I'm not an expert in these sort of things. So trying to think of what I might be able to show you that might be useful. Just to give you a case in which one thing I found useful is in sometimes using these methods forces me to conceive ideas which I thought I understood very well, the causal structure. It forces me to think in a very different way. So that's what I want to show you today. How these causal discovery algorithms force me to reimagine the generating causes of something that plant ecologists call the worldwide leaf economic spectrum. So by training, I'm an ecologist, a physiological ecologist, an evolutionary ecologist. So I'm going to be coming at it from that perspective. So again, I knew preparing this talk that most of you are from other disciplines, and so just to take a few slides just to give you a little bit of vocabulary of what evolutionary ecologists, what we talk about. So the two things important is the notion of evolutionary fitness and adaptive value. So of course, the two important people are Charles Darwin here, who gave the intuitive ideas, and Sir Ronald Fisher, who quantified this in terms of the mathematics. So the idea of evolutionary fitness is actually very simple. So if you take a set of organisms who express the same value of a trait or a set of traits, so you can do a sort of an age structure for each age. What's the probability of surviving to that age? And given that it's surviving, what are the average number of offspring that an individual will produce? And you sum over the lifespan. And this gives you something called the net reproductive output for this genotype. So this trait value in this environment, these are the average number of offspring that an organism would be produced. And when you do that, the fitness is simply defined as the net reproductive value compared to the average value over all trait values of that organism in that environment. So that's when people when we talk about evolutionary fitness, this is really just all we're talking about. And that means then if you look at different values of this trait in a particular environment, we can compare the fitness of each of these. We typically some sort of a curve like this. So those trait values which maximize fitness are those trait values which you say are the best adapted to the environment. Those trait values which have very poor fitness or producing many fewer offspring than on average is poorly adapted trait values. So the idea, of course, is that this fitness function varies with the environment. So as we begin to change the environment, these fitness functions change. So a trait value which have high fitness in one environment could be poorly adapted in the other. And so we expect then that as we look across environmental conditions, natural selection is then tracking this ridge here of maximum fitness. So we expect that those individuals which we find are most dominant should be those which have highest fitness. So that's really all we're interested in. And the important question that people who study this is, when we look at not one trait but many different traits, these traits are often very highly correlated with each other. And we want to know what is generating these patterns of correlation. To what degree is it natural selection? To what degree are these perhaps other causes independent or secondary to natural selection? Okay, so when we look at lots of different traits, we end up with something which you'll all recognize now. So this is where you get into the idea of discovery algorithms. So you have a covariance matrix or some matrix of dependencies. And we want to know then what is generating these patterns of dependencies among these different genotypes. As we change the environment, we want to know, well, one way that it could change is that the averages are changing, but the relationships between the traits, the variables, are remaining constant. So that whatever causal structure is the same causal structure occurring across environments, and you're simply changing what the average values are. Or it could be that you're changing the underlying generating causes along the environment, and we want to know which of these it might be occurring. So, really want to understand what are the generating causes here, to what degree are these can be associated with common selection pressures, to what degree can they be associated with different trade-offs between traits as you're trying to maximize fitness, and to what degree they could be other constraints. I've put physical constraints here, but we could also talk about what we call phylogenetic constraints as well. So this is the question that's interested me over many years, and this is why I became interested in these methods of causal discovery. It's extremely difficult, if not impossible, often to do manipulative experiments. You can't randomly assign size of an animal without changing all the other things as well. You can do selection experiments, but you're changing everything at the same time. So you can't really do controlled randomized experiments in this area. But we still want to understand how these traits are related to each other in terms of a causal system. So just to point out to you that when we talk about these different causes, there are different types of causes. So of course, here we have different breeds of dogs. As you all know, these have evolved between 20 and 30,000 years ago from wolves. And more importantly, if you look at the different breeds that you see here, most of this variation has occurred in the last couple of hundred years as people started selectively choosing different traits. And if you notice here, some traits are really easy to change. So size here, the difference here in a couple of hundred years is larger than the difference in the evolution of horses from the very first organisms to large horses today. And that's occurred within a couple of hundred years. Shapes of the face are very easy to change as well. So those are traits which are very easy to respond to natural selection. On the other hand, every breed of dog has only 4 legs. We can't select breeds. Some dogs have 8 legs, and others have 7, and others have 2. And the reason is that this is very strong phylogenetic constraints. These are things that develop very early during the ontogeny of the embryo. All vertebrates, they start out with these 4 limbs, and if you want to change the genes generating this very early, you basically kill the embryo, so this never occurs. So this is a very strong phylogenetic constraint. We're interested in knowing sometimes some of the correlations we see may be due to these sorts of constraints, not due to contemporary natural selection. So that's the sort of introductory part. The traits that I'm going to talk about today that I've been working on are traits related to leaves, the physiology, the morphology of leaves. So I just want to give you a little bit of an overview of how evolutionary ecologists understand how the different traits of leaves interact, and then we're going to see later on to what degree is this understanding actually agree with the patterns of covariation. So I think a good way to describe this, and this is for the economists in the field, we actually think of this as a problem of economics. So you imagine a leaf as an individual factory, the plant itself as a multinational corporation. And of course, the function of the tree is to generate profits. The function of the leaf is to generate profits for the tree. So just if you think of an economic problem, the company has to have a capital investment to construct its company, to construct its factory. Once the factory is beginning to produce, it produces a certain amount of gross income. Some of that gross income has to be repaid in order to, so if there are cars coming from this side, you have to pay money in order to get the resources, the steel and everything coming in on the other side. You also have to pay a certain amount of money just to keep pay the salaries for the workers, to repair the machines and so on. And whatever is left is the net profits. And well, we're assuming that the CAO doesn't use that to buy yachts or something, so they're investing that to maximize their income, so they're vaccinating to produce new new factories. And eventually the company has to decide that this factory has become so inefficient, it's better to knock it down, forget it, transfer whatever resources are left, than to keep it running. Okay, so that's, if you think about that, we think of the relationship between a leaf and a tree in the same way. So the plant has to invest capital in the form of carbon and nutrients to construct the leaf. So there's a capital investment that has to be recovered. Once the leaf is producing it, what it's producing are sugars, fixing carbon and converting it to sugars. Some of those sugars have to be used then to allow input of resources, carbon and mineral nutrients into the leaf. Part of the sugars have to be burned off to produce energy to maintain the leaf, to repair the various organelles and the physiology and so on of the leaf. And whatever is left over is exported from the leaf to the other growing parts of the plant, the meristems, to produce new leaves and eventually to reproduce. And as leaves age, they become less and less efficient at doing this. And eventually then, it's in the interest of the plant to allow that leaf to die, to transfer whatever resource it can to new leaves. And so the economic problem then is, how do we coordinate these different properties of the leaf? How long do you allow the leaf to live before you kill it off in order to maximize the fitness of the plant? So that's how we see this evolutionary problem. So a few years ago, 2004, this paper came out in Nature, a wonderful example of marketing that chose a wonderful title, The Worldwide Economic Spectrum, which is just a wonderful title. But basically what they did is they collected from the literature large numbers, so over 2,500 species. These are traits measured all over the world, Arctic, tropical rainforests, deserts and everything. And they measured a series of traits, physiological and morphological traits of these leaves, which relate to these different aspects of the economy of the leaf. And they wanted to know to what degree are these traits correlated, and especially to what degree do these patterns of correlation change as you change different habitats. So if you go from an Arctic tundra to a tropical forest, do you find the same patterns of correlation or do you find different patterns of correlation? I think the expectation of most people was that you would find different patterns of correlation as you change habitats because the evolutionary challenges, the selection pressures are very different. And so you should expect these patterns of covariation should be different between different habitats. In fact, they found that wasn't the case at all. So that's why they call this universal spectrum. The same patterns of correlation occur whether you're looking at an Arctic tundra, a tropical rainforest, or a desert. The correlations between the traits are the same. The average values change, but the correlations seem to be remarkably robust. So overall, the modulation of leaf traits and trait relationships by climate is surprisingly modest. And this was a surprise to people. And it was a surprise, I think it was a surprise to myself as well. So what were the variables that they measured? Well, first was a morphological variable called specific leaf mass, which is essentially the dry mass of a leaf divided by its surface area. So the idea is the surface area is the amount of energy being captured by incoming photons. The dry mass is interpreted in some ways as the amount of machinery invested to convert this energy into sugars. The second variable that was measured was the maximum photosynthetic rate. So you in the field, you put a leaf into a cuvette, you measure the change in the carbon dioxide concentration into and out of the cuvette. The difference is the amount of carbon that's being removed from the air. And if you do that under optimal conditions, highest light intensity, that's the maximum net photosynthetic rate. The third variable they measured was leaf nitrogen content. Nitrogen is the mineral element which is most commonly limiting to plant growth, and it's completely critical to the leaf functioning because it is an essential element for all enzymes, and almost all of the vast majority of the nitrogen in the leaf is associated with photosynthetic enzymes. So it's sort of a proxy of the amount of photosynthetic enzymes that the leaf is able to utilize. The respiration rate of the leaf So of course, the leaf is fixing carbon, but at the same time, it's burning carbon. These are living tissues. And so this is the leaf respiration rate. So how rapidly the metabolism of the leaf is turning over. And the final variable that they measured was leaf lifespan, the average number of days until the plant allows the leaf to die. Okay, so just remember, when I'm talking about leaf span, I'm not talking about the leaf is dying because it's being eaten. Most leaves die because the plant chooses to kill them off. Okay, so all the leaves are dying around here right now, but it's because hormonal levels in the tree are causing the leaf to die. Question?
Yes, are you including deciduous leaves?
Yes, absolutely.
Okay.
And there's an incredible amount of variation. So some species, a lifespan is 2 weeks long. There are some species where the leaves are kept for 10 years or more. In fact, there's one species in South America that the same leaf is kept for the entire life of the plant, which is over 100 years. So these are incredible degrees of variation. Okay, so just to resume, what did they find? So the very large number of species, these were data culled from the literature. They chose them from as many different habitats as they could find. The prior expectation was that the different environments would select for different patterns of covariation because the selection pressures would be different. And they found the contrary, that in fact, essentially they found the same patterns of covariation wherever you go, whether you split the data into different families of trees or whether you look at the angiosperms versus gymnosperms or whatever, the same patterns basically keep coming back. So this was kind of a surprise, and the question was then, why was that so? So well, first of all, just to give you some examples, these are all log plots. You generally have to transform these data to have linear relationships and normality. That's rather typical. So these are the sorts of patterns we're talking about. I don't know if you can see that very well. I can't even see it very well. Anyway, this is photosynthetic rate, nitrogen content, leaf mass per area, leaf lifespan, leaf mass per area, respiration rate. This is phosphorus content, nitrogen content, leaf mass per area, and so on. So you see these patterns, and these are all the data thrown in from as I said, from tropical rainforests to Arctic tundras, and they all seem to follow the same general pattern. So they didn't do any causal modeling. They didn't apply these. They simply did a principal component analysis. So this sort of summarizes what they found. So the first axis explains about 80%. And basically, you've all these variables, which are basically metabolically related variables, nitrogen, photosynthesis, respiration, phosphorus, are all highly positively correlated with each other. Leaf lifespan and this morphological variable of the leaf positively correlated, and they go in opposite directions. So they interpreted this is why they call it the economic spectrum, basically a suite of traits which are designed to conserve resources which have already been obtained, so slow metabolism, long-lived lives that don't lose their resources very rapidly. On the other hand, to resource acquisition, designed to very rapidly acquire resources, acquire fixed photosynthesis, but with very short lifespans and very thin, rapidly dying leaves. So I sort of compare this. This is the person who, when they get a little bit of money, they put it in a sock under their bed, and this is the guy who's playing the stock market all the time. And so this is sort of the range that you have between very conservative and very liberal, I guess you could maybe call it, type metabolisms. Anyways, this is how they interpret it. So what was the causal explanation that was proposed for this? The first part I'll explain it to you is basically based on ideas of natural selection. So the assumption is that the natural selection is acting to maximize the cumulative net amount of carbon fixed by the leaf per unit time, calculated over the lifespan of the leaf. So basically we're saying up to a given time, what's the total amount of accumulated carbon fixed? You take off the investment cost, you divide by how long it takes for that to get that, and we're assuming that natural selection is trying to maximize this value. And this was a model produced by a person called Kiku Izawa many years ago. And I just noticed that part of it was missing. Anyway. So part of that slide just was missing. Anyways, the result is that when you calculate this, you find out that the lifespan of the leaf should be inversely proportional to its maximum photosynthetic rate and directly proportional to this construction cost and how slowly the photosynthesis decreases over time. So if you go back to the discussion of the paper I just told you, they didn't test these ideas, but basically if you look at their paper and you translate that into a directed acyclic graph, well, it's not completely directed here. This is the explanation they gave. Photosynthesis, this is the part of Kiku Izawa's model. High photosynthesis would decrease the leaf lifespan. The cost of constructing the leaf, so if it costs more to construct the leaf, the plant has to maintain it for a longer time to maximize its carbon output. This is the morphological trait, which is basically how dense the tissues are, how thick the leaves are, and leaf nitrogen. And I modified it just slightly. So this line here comes because more nitrogen means more photosynthetic enzymes, which increases the maximum photosynthetic rate. This line here was negative because this means a leaf which has denser tissues, more carbon per leaf area, which would therefore decrease this because you would have less nitrogen per dry weight. This would increase the amount the plant has to pay to construct the leaf, which would then affect its leaf lifespan. So I modified this just slightly as you see it here, and I fully expected that this would be the explanation. This is the sort of way people understood it. And so just as an exercise, I decided to just test that as a structural equation model, and it fails miserably. That was a surprise to me. It was, I think, a surprise to other people. It wasn't completely a surprise to me because although I agreed with most of this, I had proposed a slightly different model based on the PC algorithm, actually, based on a very, very small data set many years before. And I thought that was going to work, so I was just sort of smiling when I put my model in, and it works even worse. So this was sort of an existential problem for me that, now what do I do? So I said, okay, well, we'll just give these data to the PC algorithm, and maybe they can suggest a structure which might make sense. So basically, I added to the PC algorithm. I started out with a very low significance level. Basically, it was a completely saturated, undirected graph that it gave me back. I started increasing the significance level. And you can go all the way up to 0.5, and it's always a completely saturated, undirected graph that it gives you back. So now, that was a real problem for me. I had no idea what was going on. So, an obvious explanation, although I wasn't quite sure, this must mean there's some latent variable hidden in there which is generating these covariant structures that we haven't measured. And what might it be? And I didn't really know. So I decided, okay, well, we'll use the Tetrad, vanishing tetrads to see if that can suggest. So with only 4 variables, there are 3 possible tetrad equations. It was already a saturated skeleton. So if these tetrad equations vanish, that means that it has to be a latent variable which is generating this process. So I tested that. I missed the Friday morning. So did you talk about this? Okay. This goes back to the 1973, even earlier, I suppose, method of detecting latent variables. But it's basically a simple algorithm that, given a set of 4 variables, which no pair of variables are independent, conditional on any other set, including the empty set, you test whether this tetrad equation is 0. If it isn't, you go to the next one. If a tetrad equation is 0, and if this part of it is true, then that means that there's a latent variable which is called a choke point, which is generating all the paths between those 2 measured variables pass through this choke point. So this gives you an idea of maybe where the latent variables are hiding. And when I did this, one latent variable doesn't vanish and the other two do. So that means, if I just get over to here, all causal paths linking every pair of variables except for this one that doesn't vanish, so that's the leaf lifespan and photosynthesis, pass through the same latent variable, or at least some set of latent variables, which are all going through the same choke point. That didn't help me too much, but at least it gave me an idea of what this might be. So when you look at each of these pairs of variables and you say, which pairs of variables are they being generated by some latent variable? This is what comes out of the analysis. These are the only pair of variables which apparently you can't be forced to be independent based on this latent variable. So obviously, what is this latent variable? And that's where it started forcing me to try and rethink what I thought I knew. So I'm going to explain to you how I think it's working now. And I mean, when we think about these causal processes, at least myself, I tend to forget that when we talk about different scales, we start talking about different processes. The structure which was presented was basically based on what we know about the physiology of photosynthesis at the molecular level. But we were actually looking at the level of a leaf, not at the level of rubisco molecules floating in the cytoplasm. So I thought maybe the problem is it's a question more of scale. So if we look at the structure of a leaf, I'm sure you've all seen these sorts of graphs, these sort of pictures. So this is a cell. These little things here are called chloroplasts, and this is where all the action is occurring in terms of photosynthesis. Basically, a chloroplast is a chloroplast is a chloroplast. If you go from one species to the other, there's really not much difference in their chloroplasts. What does differ is how many chloroplasts are being packed into these cells. And so maybe what was happening was that the physiology is the same within these chloroplasts. What's really differing is how you're packing these things together into bigger components. So I started to think about that. So here's a photograph. So this is one cell, and all these little dots are these chloroplasts. They're really packed in together there. And the size of a chloroplast doesn't vary very much from one cell to another. It's really how big the cell is and how many chloroplasts are in there. So this is basically, if you think of a cell, every cell of every leaf, no matter where you look at it, every vascular leaf, you have a cell wall where most of the mass is occurring. This is where all the lignin and cellulose and so on. In the middle, it is basically a big balloon full of water, cytoplasm, in which you have enzymes floating and these various organelles, mitochondria, chloroplasts, and so on. So basically, if you think that a chloroplasts are basically functioning at the same process, then what's really important is how many of these chloroplasts are in there, and that should be basically a function of the volume of the cytoplasm. Bigger cells, you have more chloroplasts. And so the photosynthetic rate per chloroplast is, as I said, very much less variable. So mostly what's generating this is the numbers of chloroplasts in the leaf, not really the molecular physiology occurring within one. The same thing, nitrogen should scale with the cytoplasm, because nitrogen is primarily photosynthetic enzymes, which are primarily within these chloroplasts. There are other components of the cell as well, but all of these things scale basically with the volume of cytoplasm. Now remember, these variables that were measured were all expressed per gram dry weight of the leaf. That was the standard way. Well, most of the dry weight of the leaf occurs in the cell walls on the outside. So it's really playing off what is the volume of cytoplasm as opposed to what is the volume occupied by the cell walls, I think, which is really generating this whole process. So photosynthesis per dry mass then should basically scale with the volume of cytoplasm within the leaf relative to the volume of cell walls. So the things that change mostly when you're looking at different leaves are the different tissue types. So some types of cells have very thick cell walls. They're primarily there for structural components. Things like fibers and factors, no cytoplasm at all inside the leaf, inside the cell. It's dead. leaves such as this have very thin cell walls. This is primarily where the photosynthesis is occurring. And the choice that the plant has is, do I produce more of one type of cell or more of another type of cell? And once the leaf has made that decision, it's basically decided how much of its volume it's allocated to one type or another. And once it does that, I think basically all the other decisions follow from that. So this ratio of the volume of cytoplasm to cell walls should determine photosynthesis per mass. It should scale positively with the amount of nitrogen per mass. The carbon cost, which is how much did it cost in terms of resources, basically should scale with dry mass. So this ratio should be inversely proportional to the construction cost of the leaf. And the more complicated one was this other variable, specific leaf mass, but it is essentially a product of the tissue density divided by the thickness of the leaf, and this tissue density is primarily determined by this. The density is the dry mass, so a leaf which has basically constructed lots of water-filled balloons would have a very high ratio. It would have a very low tissue density, and a leaf which is constructed of lots of cells with very thick cell walls have a very high tissue density and a high SLM. So this then, I think, this was the model I figured would be the most reasonable. So this structure here, if you look at it, agrees with the Tetrad equations. This arrow here comes from that theoretical expectation I told you before, maximizing natural selection, Kiku Izawa's model. And these arrows here, I think, are just, this is not a question of natural selection choosing these values. This is the only variable which I think is sensitive to natural selection. Once the leaf has chosen basically which types of tissues it's going to construct this leaf out of, and also the size of, because obviously if a large cell would have a larger volume to surface area, so that would be affecting this as well. Once you decide this, all these other parts here are basically fixed. So this is the part, this part here can respond to natural selection, and certainly you can do selection experiments and change these things without changing these other variables. The rest of it, I think, is basically rather like, you only have 4 legs on a dog. And all vertebrates have that, because this is a basic choice which you make. All leaves are constructed in this way, and once you choose this ratio, you basically have to live with whatever comes out. Now, if you test this with the structural equations model, it fits very well. It's not surprising, because the Tetrad equations told us it would in the first place. And by the way, we've tested this 2 different independent data sets as well, and this model continues to work. Is this true? Is this really what this latent variable is? I don't know. That's the next step, I guess. We can measure these things in principle. And if I can find enough of a research grant to do it, I will. But this is a lot of work. So far I can tell you that is this a model? I don't know if it's the right model. It agrees with the patterns of the Tetrad equations and so on. Makes sense to me, but beyond that, I can't tell you any more than that. But hopefully, sometime in the future, we'll be able to actually say whether this really was allowing me to detect what this latent variable might have been. So I think, yeah, that's it. So I can take some questions.
Can you think of any other descendants of that latent variable? Because if you think of 1 or 2 more, is it 2 more? You can find a tetrad constraint on the covariance matrix, and that'll tell you if it's just one latent causing all of those things.
I'm trying to think of sort of an indirect proxy that I could measure quickly. In principle, if you just measure the total volume of water in the leaf relative to the total volume of dry mass, that should be more or less proportional to this ratio. And that will allow you another descendant from this latent, which can then maybe help you to better identify that. In the data set we set there, the first one I did that, and the model considered to be working. So I said I tested with 2 different independent data sets. The second one, which we haven't published yet, is not based on different species. They're all based on different genotypes of a single species. And when we put this ratio, basically water to dry weight, if we take it out, the model works, but when I put this arrow in, it doesn't work. Is that because it's not a very good measure of the underlying latent, or is it because I haven't interpreted the latent correctly? I don't know yet. For myself, what I would prefer to is to have it more direct. So you can take leads, you can slice them, you can do image analysis and calculate these areas more directly, which would be the proper way, I think, to do it. But yeah, I've thought about that, but that's as far as I've gotten so far.
I'm trying to understand the phylogeny of this. I'm not a biologist, so deciduous versus evergreen, for example, does that figure here at all?
Yeah, sure. Both deciduous and evergreen were in here.
Oh, that was not there.
Oh, absolutely. Well, in fact, it's much wider than that. So deciduous, evergreen, angiosperms, gymnosperms, ferns, but basically all vascular. Most of the vascular clusters.
I mean, are some of those distinctions deeper down in the 4 legs versus 8 legs than others?
Of some of those variables?
Yeah.
Oh, absolutely. As I say, things like photosynthetic rate and leaf lifespan, those are things which you can select fairly rapidly. Things like the ratio of volume of cytoplasm to dry weight, I don't think so, because these are all constructed of the same types of cells. It's just different proportions of these things. So any leaf of any vascular plant is constructed in the same way. So this is a phylogenetic signal which is coming from when did the vascular plants evolve? 400 million years ago or something like that.
I have 2 questions. One is you use normal distribution tests. How normal does the data look?
When you log transform, they're linear and they're pretty normal.
My second question is how stable the linear coefficients were across your 2 samples, 2 different populations.
2 independent data sets. I have to go back. They weren't identical. The signs were.
That would be amazing.
That would be amazing. The signs were the same. I think in terms of orders of magnitude, I think they were reasonable, but I can't tell you offhand. I suspect if you put it in a multi-group model and test it, you would probably find differences between them. I would suspect.
Well, I guess the question is, you probably didn't look at this, but how much they differ compared to taking different subpopulations of this universal data set.
So it was splitting up the data set into different logical groupings.
So you said the correlations are pretty much stable across species.
Yeah.
So
I can't tell you directly, but I know that, for instance, in the original paper, if you add things like average temperature, you can find significant differences in these slopes between these different groups. They're small. There's a small signal, but it's statistically significant. It's there. So there's obviously more going on than I've talked about here.
have you published this?
What I've said printed here is what's been published. Yeah.
So this is published. Eh?
Yeah.
You got a slide with the reference?
I can give it to you. I don't have my pen. I mean, most of my colleagues think this is all voodoo, but so I'm happy when I can come here and talk to people who have some feelings for these things.
You gotta have some caution on your life.
Yeah, secret handshakes.
I take it somebody published it nonetheless, voodoo or not.
Yeah.
Do biologists generally regard this as just bizarre?
Yes. I was speaking. I can't remember you. Jeff. So Jeff is another person, another biologist. What's your opinion? My opinion is yes.
Well, on the one hand, so I do the same thing, but I work with modeling animal locomotion. Most of my colleagues, we just stick with mechanistic models. But on the other hand, there's a large number of us that just are kind of at the opposite end of voodoo, that are just use very naive association and think of everything all, multiple regression. It's all causal. So even though it's hammered in correlation, not causation, correlation, not causation, correlation, it's really hard for a human to look at these correlations and not think of them as anything other than causal. So it's sort of that weird combination. Yeah, but nobody in my field is this.
Any other questions?